And we are live. Hey, everybody. How is everybody doing tonight? Super excited. Our lecturer is not here yet, though. So I will just welcome everybody to the Central Texas Mycological Society monthly lecture series, where tonight we have Kat coming in to join us and to give us a lecture about lichens. But for those who aren't familiar with our group, we are a member self-directed nonprofit organization where we sorry, I got a little distracted with everything going on. We are a member self-directed nonprofit, which basically means that like we are kind of like a workers cooperative, but in the nonprofit sector. And when you join and you become a member, you get a vote in our organization, and you can start help directing programming and kind of get out of it what you put into it. So we encourage everybody to join through our website. Awesome. Kat is here. So yeah, memberships are, oh man, I should have looked at the website before I did this live. Now I'm all embarrassed. Memberships are $35 for a single person. No, that sounds wrong to me. But yeah, it's all on our website, centraltexasmycology.org. Membership gets you, when we are able to do in-person classes again, it gets you discounts to all our lectures. And not our lectures. The lectures are all free. That's part of what the memberships go to pay for and stuff like that. But oh, there you go. It's in the chat for me. You can just read it. I should know this, right? Okay, single memberships $25, family memberships $30, and the family is just, a little bit extra for one per house, for the household. Gets another member, kids free, and it gets you discounts to all our workshops that we do whenever it's post-COVID. Yeah, we'll be meeting in person, and yeah, and it also gets you access to our private Discord server, and which is a kind of like a place for us to come and chat, share info, share our projects that we're doing. And that's just for us members. You see, I'm a professional salesman. That's what I do for a living. Not really. That's why I'm bad at what I just did. I'll get better. I've had a long day of trimming trees. That's what I actually do for a living, and I love that. But it's a little tiring. Oh.
Wow, that participant number just keeps going up and up and up.
I know. I'm pretty excited about that.
I can't believe it. A lot of people.
If there it would be good wood for inoculation a lot of the times, but it's just the setup that I'm in right now. I can't really capture a lot of it, but it does get composted, a lot of it, or turned into mulched. No, I'm very, it's a happy surprise because I'm reading the chat for anyone who's wondering like what am I talking about? Who am I talking to? Just because we're new at doing these lectures, and it's really nice to see lots of people show up. Yeah, people are liking this event.
Was waiting for that one.
I'm liking it too.
I was waiting for it.
Yeah. Hi, Kat.
Hey everyone, sorry I'm a little late.
That's all good.
Getting set up over here.
I totally understand.
This is a great turnout. Thank you everybody. 57 people. Wow. I'm gonna try to share my screen.
You should be able to.
Okay. Okay, can everybody see what's going on?
Yes, ma'am.
Okay, and can everybody hear me okay?
Yeah.
Yes.
Cool, okay. Do you have anything you'd like to say or should I start?
Yeah, I already kind of gave a very terrible introduction for our organization and everything, but yeah, thank you so much. If you kind of want to just maybe introduce yourself a little bit, and kind of give us a little bit of your backstory, and then kind of jump into it if you would like to.
Sure, yeah. Hi everybody. Thank you for welcoming me into the Central Texas Mycology Society. I actually live in West Texas. I live in El Paso, Texas right now, and I've been here for about 4 years. So I study dryland fungi. Oops, my cat is like right here, so she might make an appearance. I study fungi in drylands or desert systems, which is the reason I moved here to the Chihuahuan Desert to study fungi in the soil, as well as those that associate with plant roots. But I do love lichens so very much. I'm glad that all of you do as well. And I actually have a little bit of an intro slide, so I can get started. But yes, I would like to say thank you especially to Allie and Angel for welcoming me in and letting me crash your book club group and inviting me to do this talk today. So thanks to you guys especially. Let's see. Okay. So a little bit about me. I grew up in upstate New York. Before I came to love the deserts of the Western United States, I was studying in the Adirondacks, going to SUNY ESF for 4 years. Then I came out west to work as a field technician on a mycology field project, which was pretty rare, pretty cool opportunity for me to move out to Utah and work throughout Wyoming and the Intermountain West, learning more about the fungi of the desert, as well as collecting lichens every day. It was such a great job for me. And that's when I really got interested in just the immense biodiversity on a micro scale that is present in lichens, as well as in other desert microhabitats. So from Northern Utah, I moved down to Salt Lake City, where I worked as a plant conservation research assistant, which was also really fun. I got to travel all throughout the state, working with rare and endangered plant species, and also started thinking about how the fungi associated with these rare species, what they might be doing, how they were helping these species persist in these really wacky habitats in southern Utah, and what nutrients they might be getting to them. So that led me into my current position, which is I'm a PhD candidate in ecology, working in a dryland biogeochemical lab. So thinking about fungi and biogeochemical cycles, how they are moving nitrogen between dryland plants, as well as working in biological soil crusts, which I'm gonna drop a little bit later. But yeah, that's a little bit about me. I like to share a little bit about my scientific journey, because even though it looks nice and linear, it actually totally wasn't. And I wasn't sure, you know, where this fieldwork would take me, but I knew I was always interested in fungi from the time I was in high school. I just thought it was such a cool group of organisms. So that brought me to where I am today. And I'm about in my 4th year of my PhD. So I have a little bit of time left to finish my dissertation here in El Paso. Okay, so lichens. Here's just a quick outline of some things I wanted to talk about today. Want to talk about some perspectives on what lichens are, what they contain, of course, the structures and symbiotic elements of lichens, the common types of lichen that you will find, some things about their biodiversity, their distribution around the world. I'm going to talk about their ecology, how they contribute to the ecosystem, as well as their really cool adaptations. Talk a little bit about using lichens as measurement tools in science. And then right at the end, just a little bit about human uses. That could be an entirely different presentation. So I didn't go super in depth with the human uses, but yeah, and I'm hoping at the end we can do questions, have a discussion. If you have questions that come up, I guess, field them through somehow. I'm totally fine with stopping and answering questions. So very cool. Okay. So I recently found this really beautifully written article in Scientific American, and it was written earlier this year, and it was called What Lichens Can Teach Us. So I wanted to pull a couple quotes from there, and also the Radical Mycology book, which is what we were reading in the CTMS book club, which kind of brought out all this interest in thinking about lichens and thinking about them from an ecosystem or a systems perspective, right? So lichens are thought to be emergent, where they're greater than the sum of their parts. That really contributes to the unique roles that they have in the ecosystem and how they are in and of themselves a micro ecosystem for all sorts of diverse microorganisms. So it's just trying to get into different ways of knowing that are equally valid as the Western science reductive way of knowing of there's a fungus and an algae, and together they make a lichen. There's so much more going on. There's so much more we have to learn about what these tiny ecosystems are doing and how those different species or taxa interact within the lichen itself. And I just think tiny things are really cool. That's why I got interested in lichens in the first place. So thinking about them from a systems perspective is something I would like to kind of push in my own scientific research, as well as just in this broader discussion about biodiversity and life on Earth. So the people interviewed in that article, one of whom was a filmmaker who actually has made a short IMAX film called Lichen. And I watched the trailer for it and it looks super cool. And I just wanted to plug that. I don't know when it will be available, but I know it was at Sundance this year. So it's just cool to think about lichens in the public sphere. And if you search news stories, there's actually a ton of really cool articles about lichens from all over the world. So there has been, there is public consciousness of lichens. They're not completely obscure and hidden like some other types of fungi that we study. But I just wanted to bring that up because I think it's really cool. And this filmmaker has a really great website as well. Okay, so getting into this, very scientific perspective on what lichens are, with all of that information, we still have some very basic constituents of what are lichens, what do they contain. So this diagram on the right sort of shows a conceptual idea of the different organisms that make up a lichen. And you can tell that there are two main components. We have the mycobiont, or the fungal partner or partners, and the photobiont, which could be an algal or cyanobacterial partner. I don't know if you can see my cursor, but in this smaller image down here, you can see how there's fungal hyphae kind of surrounding this algal cell. The algal cells are colored green. That makes sense. So the fungal partners really provide the protection and surface area of what a lichen is. So when you see it on a rock or a tree or on the soil, most of what you're seeing is made up of fungal hyphae. We're going to talk a little bit more about that too. Fungi, as we know, have a huge diversity of ecosystem roles, radiations throughout the globe of biodiversity, and so they produce a lot of unique chemicals. Almost all of those secondary metabolites that are produced in lichens come from the fungal partners. They also can produce pigments. We're going to talk more about those too. And they really protect these algal cells, especially living in these sort of harsher conditions. But the fungi and the algae or cyanobacteria both have about equal roles to play in what the lichen will look like. So the photobiont performs photosynthesis. It is an autotroph, which means it can make its own energy from the sun this like beautiful, cool part of life on Earth is that there's organisms that have chloroplasts that can photosynthesize. So that's what the photobiont is mainly doing in this lichen. And as you can see in the diagram, it's near the top surface of this lichen thallus. And that makes sense because the algae or cyanobacteria need to access light, right, to photosynthesize. So they're providing sugars in order for the fungus to persist and proliferate and grow and produce all these cool chemicals. Fungi cannot produce sugars through photosynthesis. They are not plants. They're an entirely separate kingdom. And some of you may know fungi are heterotrophs, which means basically they get their energy from something else. So they need to break down or live with, associate with autotrophs in order to get carbon. There is recent research that there's many other microbes also growing on lichens. So there's yeast that play a role in producing some of these chemicals as well. There's other bacteria, there's free living fungi, there's microfungi that live within the lichen tissues. So there's all sorts of microbial diversity going on this very, very small scale. And we're just beginning to really research that and figure out through DNA sequencing who all of the players are there. So this relationship has been described as, you know, this perfect symbiosis or this example of a mutualism, but it's sort of contested these days because the fungi really do control and change the conditions of those algal cells. It might be sort of a controlled parasitism. The relationships also change within the life cycle of the lichen. And so we're still learning a lot about it. There's still so much that we don't know. It's a complicated relationship. Okay, so because you're all fungal nerds, just like me, I just wanted to review what we're talking about when we talk about the mycobiont. So I'm sure a lot of you know what mycelium is and what an individual hypha is. So there's this underground feeding mechanism of the fungus that is the somatic living body that helps fungi procure nutrients. And the way that fungi can do that in the soil is by exuding exoenzymes to break down wood, to break down organic matter. And these hyphal networks of mycelium are constantly moving around in response to soil moisture, in response to nutrient patches. So this is in forests, and as we're learning, it's also happening in desert systems as well. And that's more of what I would like to find out, actually, how widespread are fungal networks in the soil. Okay, so when I'm talking about Hypha, fungal hyphae, this is sort of a visualization of these microstructures in a fungus. So the mycobiont represents about 17,000 taxa, fungal taxa. And like I said, most of the lichen thallus, or the body of the lichen, what you can usually see with your naked eye, that's called the thallus, is mostly made up of hyphae. It's mostly made up of fungal hyphae surrounding these cells and producing these different stratified layers. So about 1/5 of all known fungal species are lichenized. About 98% of the lichenized species are in the Ascomycota. So that is the order that also has things like morels, fairy cups, sort of the cup fungus grouping, and those are a more basal lineage or a little bit more older lineage of fungi than the Basidiomycota, which are the mushroom forming fungi. But there are some basidios that also associate in these lichenized relationships. So a really cool thing about the fungal mycobiont is that they're highly specific. So unless they're able to find specific algal partners or cyanobacterial partners with which to create this lichen symbiosis, they will not form a thallus. So they will not form the lichen body on their own when grown in culture. They're just sort of this undifferentiated mass of mycelium. They won't produce this lichen form or function without their photobionts, without their symbionts. So they need the symbionts in order to get that carbon. Some of these species, some of these taxa can live on their own. They have mechanisms for getting nutrition from outside sources. But a really important part is that they need their partner in order to form this lichen. Like, that association has to happen. So lichen classification, when identification and taxonomy is coming into play, they're mostly classified by their fungal partner because there's so much diversity in the fungi that can create lichen associations. That's what's used to identify them taxonomically. And there's a lot of fungal structures that are used in identifying lichens. And so one of those are apothecia, which are a way that fungi can sexually reproduce, create and release spores. And so when you see lichens with these tiny little cups, those are apothecia from the fungal part of the lichen. Another cool thing about fungi, we know that they're real crafty and they're always changing their life habits in order to get by. And so some can actually parasitize other lichens. Some can steal the photobiont. They can just take over those photosynthetic mechanisms and they're like, this is mine now. And so we actually see competition happen between fungi in lichen species. We see sort of this like territorial battle going on. That's another really cool and probably understudied aspect of lichen biology as well. I see there's some comments, but I can't read them. So if there's some questions, let me know.
There is one that you might kind of get into it, or maybe we could talk about it in a little bit, but it's more of a comment about the evolutionary timeline. And it's kind of like wondering maybe if it's not too much, or if you're going to get into it later, like when did lichens kind of evolve?
Okay. I had a slide on lichen evolution, but I took it out because there's like a lot of material in this presentation.
Right.
And I wasn't sure how deep we wanted to go in that, but I can talk about that as well, for sure.
Well, let's come back to that during the questions and discussion, because that's something I'm personally really curious about. So
yeah, same. It's fascinating. And there's new research that is changing those timelines as well. So
awesome.
we can talk about that at the end. Yeah. Okay. So going back to sort of the basics, the constituents of what makes up a lichen. So we have the photobiont, which is about 100 taxa of algae or cyanobacteria. So we have eukaryotic and prokaryotic organisms here. Cyanobacteria are bacterial prokaryotes. Algae are in the plant family, so they're eukaryotes. So we have some evolutionary divergence right here going on. And you can see there's way, way less taxa or species that are associated with lichens than the mycobiont, right? So one classification based on the photobiont are Chloro Lichens, and chloro is the root of chlorophyll as well. So about 86% of lichens have a green algal photobiont. So anytime you see something chloro, chlorophyll, chloroplast, that like is something green. So a lot of these have kind of been cultivated by the mycobiont over the years of evolutionary time. So we see just a few like really dominant algal taxa. One of them is this Trabuxia, and those are making up a lot of the lichens that we see in the environment. Cyano Lichens make up about 10% of lichens, and those have a cyanobacterial photobiont. And the really cool thing about cyanobacteria, we're going to get into this, is that they can fix nitrogen. So they can take nitrogen from the atmosphere that is inaccessible to living organisms and make it into an accessible form, essentially. So this is a really, really, really important ecosystem function of lichens. These Cyano Lichens, in the bottom picture here, you can see they're sort of brownish, blackish. Anytime you see a brownish, blackish lichen, it's probably a cyano lichen. So they're really important biological soil crust constituents, and early pioneers of soil surfaces are mostly cyano lichens. This top photo shows a Chloro Lichen, so it's green all over, but then there's these specialized areas called cephalodia where the cyanobacteria can live. So they can be in concert in the same lichen thallus. You can have both algae and cyanobacteria. You can have all different sorts of combinations of taxa. But this is one adaptation where the cyanos can live in a really specialized area of that thallus, where they know they're going to get light, where they know they're going to get moisture, where they know they're going to get connection to the rest of the thallus. So those are just a cool feature. If you ever see lichens that are green and hydrated and they have little black dots on them, they kind of look like little pinheads, those are cephalodia. So there's cyanobacteria that are fixing nitrogen in those little dots. Okay, so thinking to the thallus structure, again, when we think about a lichen that's kind of dry and crispy, like this photo of a Peltidra lichen, as soon as they get moisture, as soon as rainfall happens, they green up. So up here, we can see this green algal layer. The upper cortex, which is made of fungal hyphae, has become translucent in order for sunlight to get to the algal cells. So when they're turning green, the algae are photosynthesizing. So we have this green algal layer. We have this sort of fluffy inner hyphal layer called the medulla. And that's kind of making up the bulk of this thallus body. In this picture, we have a lower cortex. It's a very, very small line here. It's sort of hard to see, but those are used as well, like the size and shape of all of these parts of the thallus are used in identification. And then we also see rhizines down here, which are specialized fungal hyphae. So they're able to grow through and attach to surfaces, which is another very, very important part of what lichens do and where they can live. So yes, we'll look at those a little bit closer. This is not the most detailed picture, but this species in particular, you can see these specialized hyphae and really these rhizine or rhizoid structures underneath this thallus that are helping attach it to the ground. So that's a cool species to look for on like forest soils. Okay, so some common growth forms. This might be, some people may be familiar with this as well. But because of their needs for sunlight, for water, for moisture, lichens have diversified into different growth forms to maximize surface area, to maximize water, grabbing water out of the atmosphere. So crustose lichens are very closely attached to rock surfaces or very closely attached to their surfaces. They're usually on rocks. They would be really hard to pluck off. You would need a knife or something to like peel them off the substrate. So they're very closely attached and they usually have a very low profile. We see a lot of crustose lichens growing in high alpine areas, in desert areas. They don't have a lot of extra surface area to lose moisture in, is a big point of that particular life habit. Whereas fruticose lichens, which are up here, are more shrubby, they have a branched structure. You would see a lot of these in like a very humid, foggy, misty forest because they're maximizing water capture from the air. Folios lichens are kind of leafy. They look leaf-like. They have different lobes and sort of these articulated margins to them. You could peel them off. So unlike crustose lichens, you could just peel them off their substrate. And a lot of these are the Chloro Lichens that we see in the environment turning green when it rains or when they get wet. And then squamulose lichens are a lot of what we see in soil crusts as well. So they form these sort of different portions or squamules of structure. And that's another way to minimize water loss while also maximizing sunlight as well as nitrogen capture. So these ones are really cool in the desert as well. So where do we find lichens? Where do they grow? We can see them on wood or bark. We can see them growing on rocks. All different sorts of cool lichens grow on rocks. We can see them growing on soils. They can grow on other lichens, which is a really cool aspect I kind of talked about before, but these competitive dynamics going on. So this is like an epi lichen growing on the surface of another lichen on the on an ascus or on a apothecium. And then they grow almost everywhere. So they have really cool, unique adaptations to break down the substrate just enough to make a little tiny pocket of a happy place for them to live. We see them growing on all sorts of organic materials such as bones. They can grow on metal, on CDs, plastic, you name it. If they have the right conditions of sunlight and water, they can get nutrients from a lot of different sources. So that leads me into what are some of their adaptations? How can they live all throughout the earth? So like I said before, they rely on nutrients, moisture, and light from the ambient environment. So they do not have a root system to bring water up the way that vascular plants do. So they cannot access soil nutrients or soil water in the same way that vascular plants can. Fungi, the mycobiont excretes all sorts of chemicals outside of the lichen in order to break down some of the minerals that they need. So micronutrients, calcium, aluminum, macronutrients like phosphorus, they're able to get those from the habitats that they grow on. They can also create these really cool pigments. For all the brightest, most rainbow lichens that you see, those are basically sunscreens protecting the thallus from intense UV radiation. So you'll see a lot of those really, really bright lichens. Like this one on the bottom right is map lichen. You see that all the time in high alpine areas, places where there's lots and lots of UV radiation and sunlight exposure. So they want sunlight, but too much sunlight, too much radiation can cause mutations and actually kill cells. So that's what a lot of those pigments are doing. They're really acting as sunscreens for the lichen. They produce antibiotics, antiviral compounds to basically reduce the amount of things that would grow on top of them. They also produce chemicals to resist desiccation or to be able to survive long periods of drying out, as well as really big temperature extremes. So if you think about temperature extremes, especially in desert areas, you have really, really high temperatures during the day and low temperatures at night. So they can withstand these temperature fluxes by this huge suite of secondary chemicals that they create. Lichens can also enter cryptobiosis, which is sort of this hidden biotic state where they can really slow down their metabolism. They can almost completely turn it off and then reactivate basically when the conditions are favorable. So this means that a lot of lichens are very slow growing because they're going through these kind of stressful environmental times, but they're persisting, they're withstanding in areas where vascular plants cannot. Some of the fungal hyphae that grow in that lower cortex that are helping attach these lichens to their substrate can actually penetrate in between mineral particles. So this top picture is one that I took of an endolythic lichen. So this lichen lives its life inside a rock. And what we see on the surface are the epithecea or the fungal reproductive structures popping out once in a while when the conditions are good to throw some spores out into the air. But the hyphae associated with these lichens can access water, can access nutrients from within rocks, which I think is really cool. And then we see some lichens have these sort of root-like projections that we talked about a little bit that help attach them to the substrates. Also, they help aggregate soil particles together. And we're going to talk about the importance of that soil aggregation. But it's a really important feature of soil crust lichens, in particular, the ability to pull together and aggregate soil particles.
Okay, I have a question from the chat, and I think it goes really great at the end of this, and we might need to get into it deeper a little bit at the end. But what are your thoughts on how lichen might do as on Mars as a plant to establish O2 generation?
Yeah, that's a really good question. I mean, we know that they can survive the vacuum of space. So this little note here, there were 2 species of lichen brought up to the International Space Station, exposed to the radiation, the vacuum, the temperatures of space for 15 days, brought back to Earth. They were totally fine. They could keep photosynthesizing. Wow. So in terms of sort of this like introduced organism on other planets, I mean, they could probably do pretty well, but I think we would have to think about like what exactly the objectives are, because if you need a lot of oxygen, lichens probably aren't going to provide that.
Right. Yeah, because most of the balance of our atmosphere is maintained by photosynthesis in the ocean, as opposed to on the land. Just, I mean, that's probably as a function of the size of the oceans, but just taking up most of the planet. But anyway, back to the topic at hand.
Yeah. So, I mean, my next really cool adaptation, the take-home message about lichens, they can fix nitrogen, they can fix carbon. And those are really, really important qualities to maintain life, right? So all living things need carbon, need nitrogen in order to grow. And lichens have evolved to be able to do both of those things in one sort of like aggregated organism. So with those qualities that are pretty unique, it would be cool to send them to space, but I would definitely like, I don't know, don't shoot a bunch of like spores out into space. I'm like not advocating for that. Okay, are there any other questions before I like move on?
No, that was it.
Okay, so thinking about the biodiversity and distribution of lichens on Earth, because of all those cool adaptations that they have, they dominate about 7% of Earth's terrestrial surface, which is not a small amount. I mean, forests and grasslands and other vascular plants obviously cover more of Earth's terrestrial surface, but lichens are 7%, biocrusts are about 15%. And so with all of that carbon and nitrogen fixation, that represents a really important contribution to Earth's biogeochemical cycles. So the carbon cycle, the nitrogen cycle, these lichens are having an impact on that throughout the globe. There is about 13,500 described species of lichens. There's probably way more that we haven't described yet. The level of taxonomists and mycologists describing new species is so low in comparison to the biodiversity of our planet. So, you know, every single day, I'm sure we are all interacting with a bunch of unknown species just in our local environments, which is a really cool thing. We see a lot of species richness of lichens in environments where there's low competition from vascular plants. So it's sort of that surface area competition question. So in alpine habitats, you reach the tree line above which trees and other big plants can't survive. You can get little cushion plants, but trees that cast this big shadow or plants that have a greater surface area, wherever they are decreased because of the conditions, lichens can really thrive there. They tend to like cool habitats. They can also survive in very hot habitats, but we see the most diversity in Arctic and alpine areas of the Earth. In the coastal rainforests of Alaska alone, there's like 1,300 species described, and a lot of those are endemic, which means they're only found in that place. So they're special to that place. So if we did like a biodiversity inventory of all the like coastal Arctic rainforest, I'm sure we would discover so much species richness. As I talked about before, some lichens can also persist in about the top 2 centimeters of desert soils. And so this top picture here are lichens associated with desert biocrust. You can see it's kind of rolly and globby, and there's lichens on top. So they need to be able to access sunlight. They're not going to be found underneath the soil surface. They're going to be on top of that biocrust consortia. And then, yeah, we see a really high biodiversity in these coastal rainforest areas where there's high humidity. You can get these beautiful Folios fruticose lichens growing on branches, sort of creating this 3-dimensional structure in the interspaces of trees and plants. So the biomass that they contribute and the biodiversity they contribute is really important. It's not to be ignored. One more cool plug about biocrusts, because these are what I study, and this is what I initially got interested in. This is a site in southern Utah where we were finding a lot of endemic and endangered species. These are gypsum sand dunes, or sorry, old sand dunes that contain a lot of gypsum. And basically all of this like stuff on the ground is biocrust. So the cover, the interspace cover in drylands especially is so important for biocrusts to exist there. And so biocrusts live in the top 2 centimeters. They include lichens. They can also include mosses, free-living cyanobacteria, free-living algae, free-living fungi. They basically all come together in this top layer of soil, and it serves a lot of really important functions in drylands. And then this inset picture kind of shows you one plant popping out of this like rolling biocrust landscape. Biocrusts do have effects on seed germination, on water infiltration. They can in some ways control invasive species from germinating. There's a lot of really cool studies and a lot more research to do about biological soil crusts, but lichens make up an important part of the nitrogen fixing aspects of biocrusts especially. Okay, so into some other ecological roles. Because of all their cool adaptations, they can grow on bare rock, they can grow on bare soils. So for example, following a glacier retreat, when you really just have mineral soils, you don't have biological components to the soils, lichens can grow there. They can create their own carbon and nitrogen as long as they have water and sunlight, they can do it. Like I said before, they can consolidate soil particles, they can create soil aggregates, and that really helps with soil stability, especially in drylands where we see tons of dust storms and surface soil particles being picked up by water and dust, by water and wind, I should say. These surface soil lichens can provide erosion control. They can hold those soil particles together, hold them close to the earth so that they're not all flying away. They can actually trap dust and trap phosphorus particles in dust, which contributes a lot to the phosphorus budget of dryland soils. They can affect water infiltration. So just the roughness or the surface texture of those lichens allows water to have more places to run down into, rather than a bare soil with nothing growing on it. Water would tend to just run off, especially like a really clayey area, like a playa or something. So in that way, especially soil lichens, can contribute to preserving moisture in the soil, as well as in the ambient environment where they're growing, like in a temperate rainforest. They can capture that humidity, hold it within, and actually create an effect on that local atmosphere in terms of moisture and humidity. They cultivate really biodiverse microhabitats. Like we barely have the tiniest snapshot of the biodiversity that's present in lichens, but we already know there's several different taxonomic orders that all come together. So different lineages of life, of the tree of life, are all coming together in these sort of consortia, which I think is just really, really cool. Lichens can make up an important part of animal diets, especially ungulates. There's caribou that basically only eat reindeer lichen in the Arctic. They can get it under the snow. There's deer, mountain goats, also in these sort of more plant barren or snow covered areas. They can really rely on lichen as a source of carbohydrates. Lichens can provide nesting materials. I think this picture is very cute of a hummingbird nest. Because of the secondary metabolites in lichen, they can provide sort of an anti-herbivory protection to the trees that they grow on specifically. So if you think of a coastal rainforest, or maybe a forest near Austin that has a lot of humidity, and there's lichens just like covering the bark of trees, especially the older trees. If an insect or a bacterium or something was trying to get into the bark of those trees, having lichen there with all of these antibiotic, antiviral compounds would really stop that from happening. So they can provide this sort of protection towards the substrates they live on. And then a super important part that I'm going to drive home is the ability to fix nitrogen, especially in low nutrient habitats. So drylands and Arctic systems are thought to be nitrogen limited because there's just less that's bioavailable in those habitats. So it either has to do with the amount of nitrogen coming into the system or the nitrogen that's trapped up in the soil that plants and other organisms can't access. So when lichens are there, they can create that, they don't create nitrogen, but they make it bioavailable. And in temperate rainforests, they can represent up to 50% of the nitrogen budget. So they have a huge role to play in the nitrogen cycle all across Earth's surface. When lichens fall off of trees or start breaking down, they're still a source of nitrogen for the forest. They're a source of organic material for the forest. So we're going to talk a little bit about like collecting, but basically you don't really want to remove too much from any one habitat because they do have a really important role to play. Despite being very tiny, they have important controls on these nutrient cycles. Okay, this is a cool story. When I was working in Wyoming, I would see this vagrant lichen or tumbleweed lichen all across the soil surface. It's Xanthoparmelia, and it kind of sort of looks like reindeer lichen if you've ever seen that Cladonia species. And this lichen just reproduces asexually. It breaks into pieces. The pieces blow away. Some of them can connect to the soils and sort of form soil crusts. They can grow on rocks. It's just this really, this lichen can just like do it. It can survive on soils, on rocks in Wyoming, where it's very, very cold, very, very fluctuating conditions, especially between winter and summer. So this is a cool thing that I was seeing a lot on the soil surface, and pronghorn antelopes probably eat it. This lichen has caused massive elk die-offs, actually. So in times of starvation, these larger bodied ungulates have eaten the lichen, and because of the secondary compounds that helps this lichen persist, it was toxic to those animals. So they ate it and died. And it caused huge elk die-offs, and everyone in these sort of like, very wild animal specific areas such as Wyoming, Colorado, Montana, they started to get concerned about these lichens. It was causing a really huge impact on the sort of big game that was happening. But from all my experience with pronghorns in Wyoming, they're like not the smartest creatures. And I can see them picking some of these up, especially in a time of starvation when there's not much else for them to eat. So just an interesting story of like, kind of the double edge of lichens being able to live in many, many different places. That doesn't mean that they're tasty to everyone. Okay, so a cool way that we can use these living ecosystems is called lichenometry. So we can actually use lichen growth patterns to measure and date the ages of certain rocks or rock surfaces. We can figure out, some people can figure out how long rock surfaces have been exposed. This is not something that I've ever worked on, but it's just a really cool aspect of lichen research. So it can be used to learn more about what time glaciers retreated, so what time rocks were exposed because of glacial retreat, when volcanic eruptions may have happened and wiped out all of the primary producers in an area. When did lichens pop up? How soon were those rock surfaces exposed? As well as stuff like landslides and floods. Archaeologists can use lichens to date tombstones, rock walls, headstones, especially in places like Ireland. They use lichenometry a lot. And it's obviously variable because the growth rates are not always the same. Environmental conditions can cause a lichen to grow more slow or more fast. They've used lichenometry in places like Antarctica as well to think about glacial retreat and climatic conditions that might be allowing for rock surfaces to be more exposed for longer periods of time. Scientists can pair this with stuff like carbon dating to figure out the age of something. So at what point in time were rock cairns built, or, thinking about rock walls. It would just be a cool way to think about the impact of lichens on human history, as well as climatic history. They can also tell us about climate changes because some are really, really sensitive to air pollution. And so if you find a really hardy lichen species growing on like a tombstone, for example, then maybe all the other ones died out because of something like sulfate or sulfuric acid pollution in the air. So especially in urban areas, people have done surveys of what lichens are growing on those urban trees, what lichens are growing on those urban rock surfaces, and that can help tell us about the air pollution that's happening. And it can also give us a better picture of the air pollution that has happened as urbanization has come into these more like naturalized habitats. So there's been a lot of this work done in Europe. I just think it's a cool research direction that we could use lichens for. Okay, I see a lot more questions coming in, but I'm almost at the end and we can go through those sort of one by one. I'm really excited that you guys have so many questions. So like I said, this could be its whole own presentation about the human use for lichens. There's a long human history of using lichens in food, as spices, as medicine. There's a good section about this in the Radical Mycology book. I will say this is not my specialty, but thinking about how humans have interacted with the organisms that are ubiquitous, especially in these more northern alpine or Arctic ecosystems, is just really cool. So they can produce all sorts of metabolites, over 1,000. They have anti-cancer properties, antibiotic properties, antiviral properties. A lot of you may know that there's really cool fungi that also produce these. So it's really the mycobiont portion of the lichen that is also producing those compounds that could be beneficial to humans. and that humans no doubt have used for thousands of years. So my last note on that is to harvest responsibly. Don't peel anything off of a tree. If you find some lichen that has fallen on the ground on a road, for example, that's kind of the one that you should pick up. If there's stuff fallen in a forest, it's on the ground, like I said, it's still contributing to the nitrogen cycle of that forest. So as you remove these components, just be mindful of how much you're taking out and how that might really affect the soil organisms as well as forest regeneration. And then in places where I work in the desert, I mean, I probably wouldn't harvest anything in a great bunch because it is so slow growing, very water limited. So just harvest responsibly. This could be a whole another talk, and it would be really interesting follow-up. I have some friends that have used lichen as dye sources. There's various pigments that you can create, and they mostly work on natural materials as far as I know, such as wool and silk and stuff like that. Yeah, okay, that's the end of my talk. Here's my contact information. I'm excited to go through some of these questions right now, and hopefully you guys learned a little bit, have a little bit more appreciation for how cool lichens are. And I'm really happy that you all wanted to join me today. Okay. Let's talk about the evolutionary timeline. So I was going to have a portion of this talk where we addressed how lichens may have helped the earliest land plants come to land, because we know that there were prokaryotic organisms. We know that there were not vascular plants in those early days of life on Earth, but there had to be a way that chlorophyll containing organisms could get to the land surface, right? That's how we have this world that we live in. So it's interesting to think about fungal evolution, plant evolution, and potentially lichen-like associations, fixing nitrogen, fixing carbon, creating a stable soil environment in order for more plant species to come from the oceans on land. There has been recent research that lichen associations evolved millions of years after plants. The thing about fungi and evolution is we don't have a great fossil record. Because a lot of them are soft-bodied organisms or very ephemeral, Hyphae or Mycelium, they're not preserved as well in the fossil record. So thinking about evolutionary relationships, we really need to place them in taxonomic relation to other things that we're a little bit more sure of. So we can know when plants first came on land. We don't know what exactly was happening then. We know that Earth's climate and atmosphere was very, very different back then. There's some really cool research about early land fungi, such as prototaxites. That could be a whole nother lecture as well. But so basically, photosynthetic organisms such as algae and plants that grabbed those photosynthesis machinery, those chloroplasts, those evolved before fungi did. Lichens evolved after plants and after fungi, but we are closest on the evolutionary tree of life to fungi. So we're closer to fungal taxa than fungi are to plants. Those are completely different branches on the tree of life. So that's a really cool thing to think about as well. I am not an evolutionary biologist, so I can't give you like dates off the top of my head, but there was an article I was reading about recently that basically with new genetic information, they're able to say that lichen associations have evolved after plants and after fungi, which makes sense, but they were not there before helping plants. They were probably not there before helping plants get onto land. We don't know anything with certainty, right? You cannot prove anything with data. You can either support or not support hypotheses, right? That's the process of Western science. So the last thing I'm gonna say about evolution, I forgot. I'm gonna move on. Maybe it'll still come to me. Okay. The name of the lichen on the last slide. I see it all the time, don't know name. I don't know which lichen you're talking about, but I could go back through and like put some more labels into this presentation and that could be shared. Most of the pictures are ones that I've taken. So do lichens help make sand? I don't think so. I think sand really comes from the breakdown of mineral substrates. So when wind is blowing over surfaces, when you have wind and water erosion, that's where sand comes from, but I am not a geologist, so anybody can correct me on that. Lichens help aggregate soils. They help break down rocks, but I don't know if they are solely responsible for making sand happen, making big sand dunes happen, for example. Those are more about the mineral deposits and the other erosional factors in that area. Do all lichens produce asexually? No, they do not. I did not talk about lichen reproductive structures because it's a little bit maybe too detailed for what I was going through today, but I did talk a little bit about fungal reproductive structures. So lichens can produce both sexually and asexually, right? So asexually means that whatever they're reproducing has the exact same genetic makeup as the parent organism. Sexually means that two different cells have combined their genomes and created a new propagule that has a different genetic makeup, right? So lichens can just break off. There are certain structures where there's basically these propagule cells. Sometimes it's one algal cell wrapped up in some hyphae that can break off and fly away. And as soon as it lands somewhere hospitable, it can start growing a new thallus again. Lichens can also just release either one of those components separately. Like I said, fungal spores can come out from things like apothecia, parathecia, and algal reproduction, I don't know the particulars of, but yes, lichens can produce asexually as well as sexually. And there's definitely more details about that I didn't include. Medicinal uses for lichens, definitely. There's people that have extracted different compounds from lichens. Humans have been boiling down lichens and using them in medicine for many, 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 many decades, many thousands of years. I don't really know that many off the top of my head, but I know there's things like Litharia vulpina or the wolf lichen or usneic acid. So there's specific acids that come from specific lichen taxa that are used in medicine and that are used in medical research as well. And there's a lot of anti-cancer compounds there, but that's not my specialty either. So, okay, some programs or universities that support lichenology and lichen-related postgraduate research. That's a really good question. I don't think there are enough programs or universities supporting lichen-related postgraduate research. I was lucky enough in my undergrad to work with two fungal people, both a fungal taxonomist and a mycorrhizal ecologist. And my advisor was the fungal taxonomist, and he studied a lot about lichens. That wasn't his main specialty, but that's how I really got interested in them and started learning about them. So there are kind of more, how do I say, like environmental centered schools that would have programs where you study fungi, where you study lichens, but it really depends on the professors that are there. So I know a big hub for research is Oregon State University, OSU, because of the lichenologists that are there. ASU, Arizona State University, also has a lichenologist in their collections, who I believe teaches as well. There is the Association for Bryologists and Lichenologists, ABLS, American Association, I don't know, ABLS, you can Google that, and I'm pretty sure they have a list of different lichenologists and bryologists all throughout the country and what universities they work at. So if you're interested in doing postgraduate research, I would really recommend that you find an advisor to work with. You find somebody that is doing the thing that you want to learn about or the thing that you want to include in your research. It's really important to find the right advisor. That's my advice. Before the program, find the person. Okay, is the toxicity of the lichens determined by the species or by the environment that the lichen is inhabiting? That's a really great question. I don't know the answer to that for sure, but I think it's a combination of both. So it's both genotype, so the genetic makeup of all of these organisms that are contributing to this lichen. It's the genotype as well as the environmental factors that are influencing what's happening. So if something were growing in a really inhospitable environment, or where there's like a lot of insect attacks or something else going on, it would probably want to produce more toxic acids in order to stay living there. Because the ones that were not able to produce those toxic acids probably wouldn't have stayed alive in that space. So that's a good question. Definitely more research is needed on that, sort of looking at how genetic code is expressed based on environmental factors. That would be a really, really cool area of study for lichens. I'm sure there's people already doing that, by the way. Yep, lichens can be used medicinally. What parts of the body its constituents can support? Those are really good questions. I know there's some more literature about using lichens medicinally. I can share that with CTMS and point you in that direction. I myself actually am not an herbalist, or I don't have training, so I can't tell you the ideal parts of the body where lichen medicine can be used. But it is important, and it's really important to work in the area where you live with the people that live there and know that area, people that use traditional medicines. I would say definitely look to those resources before you go out and harvest anything. Rochester represent, cool, cool, cool. Oh, cute, 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 cute. Hi, Emily. Some of my high school friends are here. Okay, next question. Do lichens primarily contribute to the nitrogen cycle by compounds they exude, natural decomposition as they die, or via consumption, decay from other organisms? That's a good question. I would say all of the above. So they are able to fix nitrogen based on specialized organelles that they have for doing that, bringing atmospheric nitrogen into their organic material. So it's not necessarily something they exude, but those, the pieces of the lichen thallus are very nitrogen rich, and in places where there's not much nitrogen, there can be rainfall that comes through. Rain also contains nitrogen. There can be rainfall that comes through, runs through some of those lichen thalli, picks up some of those bioavailable nitrogen molecules and brings them down into the soil. So there's environmental ways that nitrogen can be cycled. Definitely when they decompose, when they die, that's a huge source of nitrogen. Like I was saying, in temperate rainforests, they make up 50% of the nitrogen budget. That's probably from lichens like falling off the trees, falling to the ground, and being decomposed. More about human interactions with lichens, totally. I could prepare something else for that. I am not the expert on that, but I kind of ran out of time to fit everything I wanted to into this presentation. Can lichens be cultivated or used in permaculture? That's a really good idea. I will respond in a way that one of my mentors, Dr. Robin Kimmerer, responded about cultivating moss in certain areas. She basically said if the conditions were right, the moss would already be growing there. So I kind of feel that way about lichens. I wouldn't recommend like taking them from one place to put them in another place. I don't think you can necessarily, well, I don't know. Maybe you could harvest some spores, cultivate some spores, like that would be a cool area of research, but I don't necessarily know the answer about lichen cultivation. It's really about getting the right conditions or the right habitat, because those propagules are all around us all the time, right? There's like billions of spores in our atmosphere coming down to where we live. We're breathing in fungal spores all the time, bacterial spores all the time. So if the conditions are right, if the habitat is right, if you can cultivate that, then probably a lichen propagule would land there and start growing. Okay, any similarity to lichens picking their algal partner and coral reef corals selecting or tolerating their algal partners? Can they change their algae over time? Yeah, I think so. I think that's a really good analogy. Coral reef corals and their symbionts. And like I said, there's not too many taxa that are involved in these lichen relationship of algae, I should say. But definitely there's a lot going on with parasitizing or switching or moving through those preferences. But some mycobionts are really, really specific, and there's some algae that just like dominate all lichens. So it was really over evolutionary time, probably the fungi are like really selecting for those algal types that they can live best with in a lichen symbiosis. I know what I was going to say about evolution now, that the lichen relationship has arisen independently about 5 times in evolutionary history. So there's not one single origin for this association. And it's an example of convergent evolution. So it's happened a number of times in Earth's evolutionary history that lichen associations have evolved. Okay, what organisms kill or decompose lichens? Or is it mostly physical or chemical conditions that do that? All of the above. I mean, we've seen that there are animals that interact. There are probably invertebrates that interact with lichens that start chewing on them, and if they can withstand all the chemicals. Obviously, they need to be broken down somehow, some way. So there are definitely fungi doing that job of decomposition. So it's a mixture of biotic and physical conditions that are decomposing lichens. Does Usnea grow in Texas? How can you grow and multiply? Yes, Usnea grows in Texas. I find it a lot in the more humid mountain areas of New Mexico. How can you grow and multiply? I don't know. Asexual reproduction is a thing, but I don't recommend like harvesting great amounts of Usnea, or it's also called old man's beard lichen, from the environment and bring it to grow in your backyard or anything like that, because in my experience, it really prefers those more high alpine areas. I mean, where I live, I'm about 3,500 feet, but in New Mexico, the mountains that I go to are like 8,000, 9,000 feet. So that's really where I find Usnea rather than around this area of the desert. Cool, cool, cool. Home DNA, PCR, and microscopy lab. For most mushrooms, we sequence the ITS gene for species identification. How is this process different for lichen, and what role can citizen science contribute to lichenology? That's such an awesome question. I also do a little bit of DNA sequencing as part of my dissertation research. I also use the ITS gene. As you may know, there are different regions of that gene that are, you use different primers to sequence, and they'll tell you different things about the taxonomic level of what you're looking at. So I think for lichen species identification, because it is really reliant on the fungal partner, you would probably do ITS sequencing. You could also do 16S sequencing to get the bacterial components. So that would tell you cyanobacteria as well as free living bacteria that are in there. There's also areas, you know, like the small subunit and the large subunit that would get you the whole spectrum of what's in a lichen probably. So there's gene regions that are more conserved over time. So by getting the sequences and figuring out the difference in that one specific sequence, we can tell how species are different, or how different taxonomic groups have changed over time and how old they might be, or how recent they might be. DNA sequencing, super cool part of what I do, the mycology research that I do. I'm gonna close out to see if I can look at all of you. Stop share. Appreciate you saying what you're not sure about. Totally. I mean, all scientists, if a scientist is like not talking about what they don't know, then there's like a lot of hubris going on there. Because the reason I was interested in science in the first place was to find out all the things that we don't know about yet and think about future directions and think about collaborations. That's the most exciting part of science for me. Just as an example, if a rock has a 4-inch diameter lichen spot, how old might it be? That's a good question. I don't know. It totally depends on the growing conditions in that area. I will say, based on personal experience, especially in this area where I live, places like Big Bend, places like Waco Tanks, so really sacred places that have rocks that were exposed a very long time ago, are full of a rainbow assembly of lichens. So thinking about how long those rocks have been exposed and how many different species have been able to land there and grow there, that's the cool thing about lichenometry for me. But saying that it has a 4-inch diameter, I mean, that's not super big, right? So it could have not been there for a very long time, or it could have been going through some really some challenging environmental variables, such as a really long drought or a really long cold season that prevented it from growing its diameter every year. Oh, I forgot to say the citizen science contributions to lichenology. iNaturalist is an awesome app. We don't have that many records for fungi in my part of the state. I don't know about your part of the state, but that's like an awesome, easy way for anyone to contribute scientific records, because when you upload those observations, you're usually uploading the GPS coordinates of where you saw that lichen, the time of day, the time of year. You can also put notes as to like what kind of tree it's growing on, that sort of thing. And if enough people verify your observation, it is scientific grade. So I know people that as part of their research, they use iNaturalist observations. And so it can tell us a lot about changes over time, changes in phenology, or the life cycles that plants and lichens go through in a year. So iNaturalist is a really cool way to make contributions to science. Other ways to contribute, I worked a lot in herbarium collections, and I worked at preparing lichens for herbarium collections. So I would say support your local herbaria. They're usually at universities, sometimes they're at museums as well, but it's really cool to volunteer there. They're unfortunately dying out, losing funding over all this time. But those collections are very, very important for scientists. And we can take pieces of lichens that someone collected in 1950. We could take a teeny little piece of it, sequence it, sequence something that we collected yesterday, and figure out what's going on with those two taxa. So getting involved in local herbaria would be really cool. You can do things like preparing specimens. So it's basically like a plant library or a lichen library or fungus library. So things get put into bookshelves, neatly sorted by name. Usually there's pictures taken. Now pretty much all herbaria have digitized their records. So scientists like me, let's say I'm looking for a specific plant species, I can actually go on this whole long list of herbarium records and figure out the exact GPS coordinates, the time of year. So it's very similar to iNaturalist, but it's like a really more permanent record of what was in a certain place at a certain time. And if you wanted to contribute some of your collections to your local herbarium, I think that would be cool too. There's certain information you want to record, but you could do it all on your phone. So that's another way that other folks who are interested in mycology and lichenology could get involved. And often the people running and working at those herbaria are kind of like the old school taxonomists, but they know so much about species and identification and using field guides specifically. So some of you might have different field guides, and in radical mycology, they have dichotomous keys. There's a lot of terminology in there that's like very confusing, in plant keys as well. So being able to work with scientists that like know exactly what this thing is, that they can show you where it is on the grass, is a really cool aspect. And I think they would be really open to having volunteers. Also museums as well. Yep, Britt Botanical Research Institute of Texas has one. I mean, there's probably a list of herbaria in this part of the country as well. Yay, thanks, Sarah. What taxonomic guides do I use? I don't spend that much time identifying lichen, to be honest. I'd like to spend more time identifying macrofungi. But these days, I've always liked the Rocky Mountain Field Guide. Common Rocky Mountain lichens. This served me really well in Utah, Wyoming, Colorado, Idaho. I don't know if I have another specific lichen guide, but there is one to the Southwest. I believe it's out of print. I looked it up yesterday and it's like $175, okay. But there are lichens of the Southwest guide as well, which would be really cool to look at. Okay, I think I reached the end of the chat questions, but please feel free to chime in. I need to rest my voice for a second.
Yeah, no worries. Thanks so much. This has been really great. We had one question from the Facebook, but my computer's been kind of acting funny on me, so I kind of lost it. But it was, if I remember correctly, it was asking, they were saying thank you for the good information on sequencing, but they were wondering if you had any book recommendations for that. more in-depth reading on that maybe.
Book recommendations for sequencing fungi.
Yes. Or
Um,
just maybe sequencing in general. Maybe if they're still watching, they could ask the question again. And
off the top of my head, I can't really think of a book.
Really?
There's a lot of good papers out there. There's a lot of papers that bring it through step by step. There's a lot of open access information about sequencing, and there's a lot of websites. I can't think of a book off the top of my head that I use for sequencing. There are various textbooks, like I have one, it's actually holding up my computer, LOL. Like something like this, which just basically is like a compendium of different research papers, different articles, all brought into like a textbook format. So there's stuff like that that has sequencing information, but I would go on Google Scholar, search fungal sequencing, search fungal ITS gene region, and see what's open access that comes up, and stuff that there's some classic papers, like people that identified that gene region in the first place, people that are creating primer sets for that gene region, really pushing that forward. So yeah, that's a good question. I'll try to think of some if I can. And CTMS, we have like a book club resource Google Doc, which I think is open to everybody. And I've been putting some stuff in there as well, like news articles, some journal articles, some videos, stuff like that. So if you guys have access to that Google Doc, there will be some resources in there about lichens.
someone mentioned that there's a fungal sequencing group on Facebook. That's really helpful.
Yes, yeah, I'm a part of that group, and they have great experience doing at-home sequencing setups, which I've never had the privilege of setting anything up at my house except a microscope. So I've always been able to access like a research lab to do sequencing, but you can do it at your house.
And I just wanted to add, I have to make that Google document public to everyone. Right now it's just for the people in the book club, but I can totally put it on our website.
And that's super cool.
Best place.
Cool. Thanks, Allie. I saw a question about what's my favorite lichen. Oh, that's so hard. I really like Rhizocarpum geographicum. I really like the map lichen because it's been to space. It's just like really cool and distinctive to me. And it was one of the first ones that was like an easy ID when I was coming out west, especially in the Rocky Mountains. It's everywhere. I also really like the wolf lichen, Lotharia vulpina, that's super bright lemon neon green. It grows all over trees in like Yellowstone area in Wyoming. It's super beautiful if you can find it in large amounts. So those two are really cool, but I love all lichens. It's like, don't make me pick just one.
Yeah, I really like I'm drawing a blank on the name. I think it's the Folios ones, the ones that are kind of stringier, and the Usneas, I think, are a type of that one. Those are my favorite because they're like, I don't know, they're just so weird, and they're, you get the rubbery, you pull on them and stuff like that.
Totally. Also fairy cups. I love any and all Cladonia species. They're so cute, and they make these like adorable micro worlds on like a piece of bark or a rock. And they often grow with really beautiful mosses as well. So I like all the fairy cup lichens or Cladonia lichens. Those are some favorites too.
Yeah, I just love it when it rains and they make the spore carps, and they start to like put out the little cups on them and everything, and then there's the ones around here that are really bright orange in the middle of all the green. That's really cool too. Let's see, there's a question that says, do you know the orange lichen on the West Coast that indicates clean air? Do you know which one that is? Like, I guess the name?
There's a lot of orange lichens, but maybe something like Xanthoria or what's the other one? Caloplaca. Those are two orange lichens that I know off the top of my head. I'm not sure what the air quality response is, but I think that's such a cool field of study. I don't have enough information off the top of my head on air quality monitoring with lichens, but You could probably Google it. I can write in the chat what those genera are.
Do we have any other questions? Maybe from the Facebook audience or the YouTube people?
I have a question. Is there like a, you know, how in the mycology world, or like there's certain mushrooms that like captivate like the popular culture, or like, you know, people that sort of dip into like an area of the natural world, like it becomes like a real like fascination. Does lichens have a species that people really like lust after, or like they want to find it?
You would have to ask like an actual lichenologist about that, not just like a lichen enthusiast. One that I can think of that's really cool is lobaria, or the lung lichens. They're really just like fascinating and charismatic, and they grow in like the Pacific Northwest. They grow in these really humid forest areas, and they have some interesting properties as well. So that's one that people might be interested to like hunt for and like take a picture of. Hopefully not.
Not destroy. Yeah.
Yeah, quick tips on lichen microscopy. That's a really good question. So usually when I'm hiking, I carry a loop, which is like a little handheld kind of magnifying glass that scientists use. And that's a really cool way to just get like a quick look at something that is so tiny. And you can be like, oh, there's apothecia here, or, you know, there's color change happening. In terms of looking at it under a compound microscope, definitely it's like make the thinnest section that you possibly can. As with anything in microscopy, compound microscopy, you just want to make it like the absolute thinnest one cell layer look that you can get, so that light can come through it, can come through the slide, so you can see the structures inside. So practice with making sections using a really sharp razor blade, and that's my main advice. There's also some cool color changes that you can look at under the microscope with stuff like KOH, some of the stuff you could use for mushroom ID. Yeah, you can use reagents. You can use the same reagents. I don't have like extensive knowledge, but I know KOH would be one of them, and probably, I don't know, potassium chloride, one of the other ones. And they'll change purple or darker. You can do that on a microscope. It's really cool.
Awesome. This is such a fascinating subject. Really appreciate you coming out and talking with us tonight.
Yeah, thanks for letting me crash your group. I'm down to start a West Texas chapter. That's all I'm saying.
Awesome. Well, yeah, I think organizing this group kind of took a turn whenever COVID happened, and the internet stuff was always kind of something that we wanted to do, but it was kind of something we kind of put on the back burner just because like it made more sense to like try and get people in person and meet up and actually like go on more walks and things like that. But you know, now it kind of afforded us the opportunity to have talks with people from that where we're not bound by geography. So that's pretty fun too, and it's something I definitely want to keep up into the future. So
Yeah, I think it's great. I would love to come out to Central Texas and do some forays with you guys once everything has passed. Maybe next summer.
yeah, next summer probably. Ugh. But we'll have plenty more meetings like this and more recordings and whatnot. Yeah, we started doing these Myco hangouts on Mondays, or not on Mondays, on the first Thursdays of every month, where it's like a little interview that we do, and it's this little similar format, but it's a little more casual. And we're gonna even try and make it even more casual in going into the future. So
Yeah, I would love to do a follow-up presentation and do some more research into all these awesome questions. My semester starts in a couple days, so I don't know when I will be able to do that, but maybe in the winter. This is a really fun part of being a scientist, by the way, is like just talking with people, fielding questions, getting people excited about what I'm excited about. I mean, that's like my favorite part. Being in the lab by myself, not my favorite part.
right, yeah. Well, and I know Angel's gonna kind of talk a little bit more about this here in a second as we wrap up. But yeah, that's definitely one of what we want to try and do, is be like a bridge between people who are
Mhm.
alone in the lab all the time and kind of give them that social outlet with some people who are interested in, and hopefully, and now like we're doing it sans geography, so we're creating our own virtual spaces, which I think is kind of cool, and we're gonna push this as far as we can go, this medium as far as we can, and hopefully, I know I'm trying to up production quality a little bit so it'll be real slick for the interwebs.
Yeah, so like Philip said, we'll have a little bit more programming. We're doing a little bit more online activity, and we'll be doing a myco hangout with Smugtown Mushrooms from, I believe she's up in New York State. Is that right, Phil?
Upstate New York.
Rochester, New York.
Yeah, New York represent upstate.
Do you know Olga? Kat?
We've never met in person, but I know
Oh, cool.
her. Yeah, she's definitely Rochester famous.
So yeah, so
I think
we
she's
invite
here
everybody-
actually.
Hey, Olga.
She's in the chat. She says hi. Yeah, it's gonna be
Hey.
fun. We're excited. We're gonna be talking about the tours that she does, where the mushrooms of Greece, and a little bit about Smugtown mushrooms, and her business and how she got started with that. And we'll also be talking about why psychedelics and psychedelic mushrooms aren't going to be enough to stop the rise of fascism in the United States. So it should be pretty fun talk, covering a lot of different subjects.
No band-aids.
No band-aids, yeah.
Yeah. So if you all want to keep up with everything we have going on, you can follow us on social media. If you're not a social media person, you can always get our newsletter. We keep up with our events. You'll get a couple emails. We won't spam you too much, but we do some giveaways. We work with a lot of local businesses within Central Texas that are cultivating mushrooms, doing something within the fungi kingdom to continue to spread the spores. So there's a lot of ways to keep in touch. So yeah, we thank everybody for coming out, and yeah, this was an excellent presentation. Thank you, Kat, so much, and it's going to be fun to continue to go through radical mycology with you. We have a few chapters left, and then if anybody wants to join our book club, we meet twice a month. You can email us. I think our email link is on our website in the footer. And our next book that we've announced is the new book that came out from
Merlin Sheldrake.
Entangled
Life.
Entangled Life.
Yeah. Called. Yeah.
So if you're interested in having another social group online, we'll be reading that book next, and it'll probably be starting late September. So yeah, you can still sign up to be on our email for the book club as well.
Yeah.
All right.
Awesome.
Is that it? You want to take us out, Phil?
Yeah. Thanks for coming, everybody. I'm gonna just go ahead and end the